Welcome and thank you for standing by. At this time, all participants are in a listen-only mode. During the Q&A session, if you'd like to ask a question, you may press star one on your phone. Today's call is being recorded. If you have any objections, please disconnect at this time. I'd like to turn the call over to Ms. Irene Ahir. You may begin.
Thank you. Hello, this is Irene Ahir of CDRH's Office of Communication and Education. Welcome to the FDA's sixth in a series of virtual town hall meetings to help answer technical questions about the development and validation of tests for SARS-CoV-2 during the public health emergency. Today, Timothy Stenzel, Director of the Office of In Vitro Diagnostics and Radiological Health in the Office of Product Evaluation and Quality, and Sarah Brenner, Associate Director for Medical Affairs, both from CDRH, will provide a brief update. Following opening remarks, we will open the line for your questions related to today's discussion. Now, I give you Timothy.
Hello, thanks for joining us again. I hope everybody's finding these town hall meetings productive. I look forward to another one today. We have many updates, but it won't take me long, I think, to go through some of them. So we have many new EUAs authorized since last week, including 4 new serology authorizations. Please check our FDA EUA authorizations webpage. Also, we've made numerous updates in the last week to the frequently asked questions, including a new extraction option, so that hopefully is welcome news. Finally, an important update is that we have a new serology pathway that was just set up last night. It is called, we called an umbrella pathway for serology. This, at a high level, applies to serology tests that are submitted to the interagency test group. Currently, testing is being performed at NCI. And those companies that submit their serology, mostly rapid tests, but some ELISAs are able to come through this pathway. To this program, testing is performed currently at NCI, and then if performance metrics are met, then those tests have a pathway to authorization under this umbrella pathway. So I will speak to a little bit to the specifics of this pathway. If it's not publicly available yet, it will shortly be publicly available on our website. So this is for the devices that include in vitro diagnostics, SARS-CoV-2 antibody tests, lateral flow or enzyme-linked immunoabsorbent assays, ELISA tests, that have been evaluated in an independent validation study performed at the NIH's NCI or another government agency designated by the FDA, and are confirmed by the FDA to meet the criteria set forth in the scope of this authorization letter. Let me go through what those performance metrics are. All right, as performed by the NCI or another government agency designated by the FDA, using a well-characterized panel of at least 30 confirmed SARS-CoV-2 antibody positive for each, and 30 for each immunoglobulin the test is intended to detect. Currently, we're restricting this testing to IgM and IgG devices. Mostly there are Ig only or pan, or those that distinguish between IgM and IgG. These are serum and plasma samples, and then 80 antibody negative and or pre-COVID-19 serum plasma samples. 10 of the 80 negative samples must be confirmed to be HIV positive. The data must demonstrate a minimum overall 90% positive percent agreement, otherwise commonly thought of as sensitivity, and an overall 95% negative percent agreement, or otherwise normally considered specificity for the tests that report specifically IgM and IgG results, or have the ability to report a PAN result. A minimum of positive agreement for those tests that test for and report out IgM and or IgG is that for IgM, there must be a minimum of 70% positive percent agreement or sensitivity for IgM, and a minimum positive percent agreement for IgG of 90%. The data must also demonstrate no cross-reactivity with HIV in the testing. So, so this program is now available to developers, and they can approach us by emailing us at the templates address. And with that, I concluded my opening remarks and comments. And unless Sarah has something, we'll open it up for questions. Thank you.
Thanks, Tim. This is Sarah. I'll just remind folks that last week I discussed the lab data harmonization effort under a project called SHIELD at FDA in coordination with CDC and other federal partners. I believe there's a slide that is either up or will be up on the WebEx providing that email address for more information. That's it.
The phone lines are now open for questions. If you would like to ask a question over the phone, please press *1 and record your name. If you'd like to withdraw your question, press *2. One moment for the first question. There's a question in the queue from Brant Midler. Your line is now open.
Hello, Dr. Stenzel. Thank you for providing this opportunity. I think there was a question last week about a protocol for point-of-care testing with, say, rapid diagnostic test kits. And the question had to do with whether you all wanted just demonstration with the ability to, follow instructions, open the materials, follow the IFU, a survey perhaps, or if it had to demonstrate the actual ability to get a true positive with a sample or a true negative. Could you expand a little on that? Since I think you also commented about, demonstrating that the person doing it, say in this case a trained physician, would be equal to a trained laboratorian, I think is the term you use. So I'm talking specifically now about these rapid serology diagnostic test kits.
Yes, so in general, for a point-of-care type authorization, we will ask for lay user studies. These are studies to demonstrate that a lay user can get accurate results when performed in that kind of setting, under those kind of conditions, with the kind of instructions that are provided with the test itself. And ideally, those would check for accuracy of the ability of the lay users to perform that testing. That is a required part of any new device that we haven't seen before to perform, that we haven't previously authorized for a point-of-care setting, for the ability to be designated as such as a point-of-care device. I would also like to add that we've been working hard on serology templates, and they are very close to being done, and the expectation is that they will be out at latest the end of this week, but maybe sooner.
Okay, just one clarification. You talked about lay users. So what does that mean in terms of a physician using it at point of care outside of a CLIA complex lab, say under a study under an IRB? Is that appropriate to define point of care in that way?
So we have some broad language that covers near patient point of care, and our FAQ page for point of care describes many of these. And I would have you refer to our frequently asked questions page for those environments. When we say lay user, we are expecting that it's a healthcare professional of some sort, but it's what we're demonstrating, or we're asking the developer to demonstrate, is that a non-laboratory person, somebody who isn't trained in laboratory procedures, but maybe in a setting, in a clinic where they don't normally perform tests as part of their all day long, everyday activities, that kind of user can get an accurate result with such a device. And so it involves clear instructions, easy to do, easy to interpret, performance expectations.
Thank you.
Next question.
Next question is from William Chop. Your line is now open.
Yeah, this is Bill Chop from a lab here in Grand Rapids. My question is kind of a general question. It deals with SARS-CoV-2 PCR testing of asymptomatic individuals by nasopharyngeal collection in non-healthcare settings. Just wanted to see what the FDA stance was on that, either a like a workplace setting, collection by a healthcare individual, or the supervision of collection by an individual by a healthcare provider.
So where would the test actually be performed?
The test would actually be performed within a CLIA certified lab. Just the collection process would be outside of there in asymptomatic individuals.
Okay, so as far as the collection by a healthcare professional, I'm not aware of any FDA regulations that have stipulate when a healthcare professional is performing the swab. We obviously have authorized self-collection, but not for a nasopharyngeal swab. You specifically also asked about asymptomatic individuals.
Yes.
So currently we have not authorized an EUA test that has claims for detection of SARS-CoV-2 molecular test for asymptomatic individuals. If a manufacturer or a laboratory wishes to make a claim about the ability to accurately detect SARS-CoV-2 in an asymptomatic population, that would require an EUA submission for that specific claim and a study powered well enough to demonstrate the capability of detecting an asymptomatic person. So, and we don't exactly know what the asymptomatic carrier prevalence is out there, so it's really hard to know how many people you might have to test in order to detect that. However, if you're asking, can you use in a given EUA authorized molecular test, take a nasopharyngeal swab and perform testing on an asymptomatic patient? That is not something that we're going to say, what's the best way to put it? This is, I think, falls best under a currently listed frequently asked question, where we have said that laboratories that receive samples that aren't, I forget the terminology, but aren't something that they have specifically validated, they can receive those samples, perform the testing, but they can also amend the report saying, if they happen to know that it's an asymptomatic individual, saying that their test hasn't been validated for an asymptomatic patient and that a negative result does not mean they don't have SARS-CoV-2. So again, I think the important thing is if a developer or it's a lab or a manufacturer wants to claim an asymptomatic sample type, that would require an EUA authorization.
Perfect. Thank you for your time.
Uh-huh.
And again, if you would like to ask a question over the phone, please press *1 and record your name. And also, please limit yourself to one question. Thank you. Steve Skaggs, your line is now open.
Hi, yes, good afternoon. I had one question about the umbrella pathway that you mentioned. The NCI is going to be performing those tests. I believe you mentioned this, but I want to make sure the best avenue to get information about getting in that umbrella pathway is through the templates email. Is that correct, Tim? And you mentioned having a website open or something of that nature aside from the templates. Is that correct? Thank you for allowing that.
Yeah. So, yeah, sure. So you can ask to be part of that program by emailing the templates address. You'll be connected to that program, and you will receive information about ship to and the number of tests that would be shipped to the location. The umbrella pathway is in the process, if it hasn't already been posted on the FDA website, it's in the process of being posted. So keep attention to the website to see all the details in this new pathway. And then the last question, what was the last part of that question?
You got it, actually. It was, I asked if that was going to be a separate website or part of through the template, and I think you just explained it, that it will be noticed on the FDA website. So thank you very much.
And then in addition, there are more general serology templates that will be posted in the near future, hopefully by the end of the week, but hopefully sooner than that.
Great to hear. Thank you.
Next question is from Sam Melmy. Your line is now open.
Yeah, I'm hoping you can share some perspective on the unintended consequences where Pathway D manufacturers can sell easily in the U.S. and have them administered by point of care, granted without verification. But once someone gets an EUA, then suddenly they are required to be performed only in CLIA labs, and they are also subject to the restrictions of reporting back to the FDA who's using them, false positives and false negatives, so that the companies with an actual EUA end up having most of their product be bought by foreign governments who are willing to use them at point of care, utilizing our gold standard FDA EUA. Thank you.
So hang on, I want to make sure I'm answering your questions. So first of all, those who notify us via Pathway D are only deemed to be used in the U.S. in high complexity labs. They are not to be used in moderate complex labs or in point of care or home use in any way. In order to get those that authorization, they must come in for an EUA authorization. Also on Pathway D, those that have just notified us and have not been authorized, they are formally not FDA authorized. And let's see, you had some other questions there. These are deep and, point of care. What is, I just want to comment on what it takes to be point of care. So if they want to be moderately complex or point of care, they come in, they demonstrate the performance in those settings, and we can deem those as point of care. And then they can be used in the point of care setting. But those, not authorized for those settings, and those that haven't notified us cannot be in the U.S. right now for those purposes.
So my understanding is there's no enforcement mechanism for Pathway D. They can sell directly to point of care providers, and they haven't necessarily covenanted that they're not going to, whereas anyone with an EUA has in their EUA specifically covenanted that they're not going to be used outside of complex laboratories and that they're going to report back everything to the FDA. So the restrictions on the EUA tests make them less attractive in the local market from a practical standpoint than those with an actual EUA.
So those that have simply notified us that they've validated their test through Pathway D, they are allowed under a high complexity lab certificate if that lab sets up, say, a draw station. That lab is allowed to do, under their high complexity CLIA certificate, under their rules, to do, quote, point of care testing. But if someone has simply notified us, a developer has notified us through Pathway D, and they are selling into a point of care setting, that is not allowed. It's not authorized. We have a fraud email on our FDA website, EUA website, that you can send information about that, and we will investigate and take action as appropriate. And if that email address is not easily available, you can always send something to our templates email address. But if a developer is selling something into the point of care testing world, not under a high complexity situation, they are not authorized to do that. We will investigate and we will take action as appropriate. Okay, next question.
Next question is from Daniel. Your line is now open.
Hi, thanks, Tim. Appreciate the weekly public information sharing session that you guys are putting. I have a two-part question. One is, what do you envision as a path forward in U.S. for use of serology test kits? Currently, it's only accessible to use at high complexity labs. Some developed countries have gone as far as making these available for home use, if I understand correctly. When do you see FDA policy changing to allow utilization of these tests at least point of care level? That uncertainty from a commercial standpoint, in my opinion, could reduce the R&D effort and development or investment activities for these test kits. So that's part one. The part two is we've seen so many unverifiable claims by suppliers of serological tests, companies claiming their FDA EUA application or U.S. distribution notification has been submitted or is in progress. In spirit of transparency, could FDA consider maybe publishing the status of developers that have applied to EUA? Of course, we can see the approved ones, but more, I guess, related to the ones that are in progress or have been rejected, so that we know which manufacturers or suppliers to focus on or consider working with. Thanks.
Okay, so the two questions are, I think, point of care, the need for that, and status of submissions of serology tests. So we do make the serology status of tests that have been authorized clear, that have notified us but haven't been authorized clear. We also have now made clear on both the authorized and the notification pathways what clinical environment they can be tested in, whether it's high complexity, moderate complexity, or point of care, or what we call waived with the W, so H, M, or W. It is more challenging to provide updates other than that, because that is oftentimes considered proprietary or confidential information. If a test is denied authorization, if under the NCI umbrella pathway that I mentioned earlier, if they do not meet the performance bar, they will be removed from the notified list, and potential other further actions may be taken. So that those tests that we deny in some way will be removed from the pathway notified list, and that will be one way that we make this information public as soon as we make that decision. Hope that answered your questions.
That did, and I guess maybe about the first part of the question, what's the path forward, maybe as far as allowing point of care?
Yeah, so the pathway, if you come to us through our EUA templates address and ask what is required for a point of care validation, we will provide that to you. We are in the final stages of drafting the serology template, which will lay out these things a little bit more clearly for all to see, and will be posted publicly when they are signed off.
Thanks.
So our policy is that we will, and you said also homes, right? Our policy is that we will authorize point of care serology and other point of care tests if they meet the performance expectations in working with our reviewers for those settings. So there's no prohibition about that. There's also no prohibition about home collection or home testing. It's just that in the home collection, home testing situations, those require EUA authorizations. They require specific accuracy tests to be done so that we know in those environments accurate results can be obtained. And the same goes for the point of care setting. So it is policy that we will authorize these if the correct studies are performed and the accuracy is there that we can authorize them.
Excellent. Thank you.
Mm-hmm. Uh-huh.
Next question is from Erica Tyberski. Your line is now open.
Thank you. In regards to home testing, agnostic of type of, type of sample, what would be the gold standard we should be comparing against for performance characteristics?
So what kind of test are you developing? Because it depends, whether it's a molecular, whether it's a serology, or whether it's a direct antigen test. So sort of at a high level, the performance expectations are the same as if they would be performed in a high complexity lab. In addition, we require for point of care a lay user study, or for home use a consumer study that demonstrates that in each of those other environments, accurate results can still be obtained.
Okay. Thank you.
Mm-hmm.
Next question is from Sarah. Your line is now open.
Hi, Sarah.
Next question is from Alberto Gandia. Your line is now open.
Hi. The question is, has the FDA start considering any antibody titer test? That's number one. And then in any of those pathways to get the AUA approved, is the test need to be performed anyway with finger prick, or that is not being considered? It's more part of a point of care for home testing that will be, in my understanding, it will be done later.
So two questions. One is titer-based tests, and the other is finger stick tests.
Yeah.
Good questions. I believe we already authorized a lab-developed test, a laboratory test at Mount Sinai that does a titer determination. So absolutely, we will consider authorization of a titer test. Obviously, that has different performance features that we want to make sure are accurate. So engage with our reviewers at the EUA templates site right now. The templates that we're working on now will not specifically show what suggested or recommended development test should be for titer or semi-quantitative or quantitative serology tests. So anything other than a qualitative test, engage with our reviewers on what would be required for that. And then finger stick. So the templates will lay out a recommended pathway for demonstration of performance accuracy with the finger stick. It could be compared to another sample type that's been fully validated, like serum, plasma, or venipuncture whole blood.
Yeah. Thank you.
You're welcome.
Next question is from Kay Jewell. Your line is now open.
Hello. Thank you for all of this information. My question is about the umbrella pathway. In terms of what the cross-reactivity it's requiring a HIV, will that replace, will they still also have to do the cross-reactivity and the class specificity that's addressed in the March 16th policy?
So the classic or the other already open pathway for serological testing is still open. This new umbrella is a pathway that goes through NCI or other government agency doing testing on 30 positives and 80 negative samples. 10 of those negative samples will be HIV positive. And that's because there is some concern of potential cross-reactivity in HIV positive samples. So those were specifically added to look at that. We assess that if performance is good on those 80 negative samples, basically above about 98%, no specific additional cross-reactivity testing may need to be performed with specific potential cross-reacting agents. However, if in the usual pathway, if performance falls below a level, we would ask for additional cross-reactivity testing to determine the reason why the specificity is lower than expected.
Thank you. Thank you very much.
Mm-hmm. Mm-hmm.
Next question is from Mark Manulo. Your line is now open.
Yes. These are terrific conferences. Thank you. Wondering if you can detail some of the specifics regarding the serology tests, in particular potential cross-reactivities with other coronavirus species, or sort of what the threshold is there.
Well, at the very minimum, if any cross-reactivity is detected, it would have to be in the instructions for use in the performance section. If it reaches a certain level, number of cross-reacting samples, that may impact the potential for a positive authorization. So it's
well, I guess a more maybe simply put is if are the tests that are examined, are there panels of sera from other non-COVID-19 infected individuals that are used in the screening process and as a parameter of either approval or not?
There is not any sort of FDA-sanctioned panel, commercially available panel at this time. It's hoped that could be happening. I've asked, and I haven't checked whether our FAQ page lists potential providers of validation panels. For those providers of such panels, we have a process by which we vet those firms, and if we feel it's something that we can make widely available to developers as far as information goes, that you can go to these providers, we will do that. I apologize, I haven't checked that list lately. We have had some providers approach us, and I've asked that they be reviewed, and as soon as we can post anyone on the FAQs, if we haven't already, we would post them. However, we do know some providers, and if you email us at EUA templates, we can at least give you contact information, and since if it's not on our FAQ page, it would basically be information without any guarantee that at this moment that we've taken a look at them and we know that that's a good provider. But I know that there are providers out there that are developing these panels, and as soon as possible, we'll make as many of them as possible, place them on our FAQ page. And if we get to the point where we can specifically recommend something or sanction something, we will do that. We understand that it is challenging to get these materials.
Terrific. Thank you.
Next question is from Ted Liz. Your line is now open.
Hi, yes, thank you. Good afternoon. My question is for Dr. Brenner, if she's still available, or Tim, if you could answer. At a town hall,
she's probably better at answering almost anything. So,
all right. At a town hall 2 weeks ago, it was mentioned that FDA is preparing recommendations pertaining to 3D printing of nasal pharyngeal swabs. I was wondering whether we will be seeing that document soon, or if it's still in progress. And specifically, during this public health emergency, will FDA expect the hospital to register, list, and pay fees as a manufacturer if it prints the swabs only for the internal hospital use of its own patients?
Yeah.
Sarah, are you?
Yeah, Tim, I can take those if you want, and feel free to.
Go ahead, sir.
Sorry, I went from one connection to another. I'm back. So those are great questions, and I know we have discussed the issue of 3D printed nasopharyngeal swabs and other types of swabs on this town hall call for many weeks now. So I thank everyone for their patience. What I can share at this point is we most certainly are preparing some sort of information or guidance. I don't want to be too prescriptive about what form it will take, but we are working on what I believe the community will find to be very informative based on the questions that have come in so far. And then there's a process that we go through internally to have that verbiage vetted and cleared. It will probably be out of our office this week and moving up through the necessary channels with posting hopefully as early as next week. I'm not making that promise, but we've certainly been striving to get all of the information put together and presented to the public as quickly as we can, because I know there are many, many questions and many people trying to address shortages through these very creative manufacturing means. So that's one thing I'll say. The other two other points on that, one is that we do intend to have an open discussion, a town hall-like forum where folks who are interested in a dialogue with FDA and other federal partners can join in sort of an open dialogue. And those of you who have reached out by email following these town halls would certainly be welcome, and any others would be welcome as well. So we're shooting to get that conversation stood up and broadcast to appropriate parties at some point next week. Again, thanks for your patience and stay tuned. The last piece I'll mention related to that is our internal team, which includes subject matter experts, policy folks, regulatory experts, legal folks, the whole gamut. We've been following the literature closely, including data that has been submitted to us for analysis and data that's been submitted to federal partners, including NIH and VA, who, as I believe most folks know, we have an MOU or partnership with on 3D printing and additive manufacturing. And so there are many experts across FDA and our agency partners who are trying to stay ahead of the curve with regards to what's being manufactured and advertised and marketed and used out in the field. So in coordinating with those folks and stakeholders that have reached out to FDA and other parts of the government, you know, we're looking for a sort of harmonized way that we can all work together safely and get products out there that are going to be effective and safe, and then also where that fits into our regulatory framework, given that these have traditionally been Class 1 exempt devices. To your question about registration and listing, I'm not sure that we have a definitive answer on that yet, but it is something that will go into the guidance or information that we put out. We definitely are aware of that issue and that question.
Thank you for your answer, and I really appreciate all the work you and everyone at FDA have been doing and for these town halls. Thank you so much.
Mm-hmm. Next question is from Lalita Kota. Your line is now open.
Hi, Dr. Stenzel. Thank you so much for your time and answering all these questions. They've been really helpful. I just wanted to know, what is the FDA recommendation on reporting quantitative results for serological tests for IgG and IgM?
Quantitative results for IgM and IgG. So, Tim, first of all, just thank you. You know, this is a question best left for our expert review staff. I know that, you know, a straight sort of titer positivity determination is pretty straightforward. But if you're going to do quantitative testing, there are some additional considerations in development and performance evaluation of those, and I would defer to them. So our templates address is a perfect way to ask that question.
Okay, thank you.
Mm-hmm.
Next question is from Sarah. Your line is now open.
Hi, good afternoon. Can you hear me?
Yes.
Hi, thank you for holding these town halls and describing the new umbrella policy. I just have a general question about Pathway D and the serology test. Is FDA considering that all of these tests will either need an EUA in the future or some other FDA approval besides just the notification?
So that that consideration is being thought about right now, and there may be something in the works in the near future to address that specific question. So I would have to defer until that those deliberations are completed and we're able to make something public about that. We do understand the concern out there about at least some of these tests through Pathway D.
Completely understand. Thank you. So just one quick follow-up. So are you suggesting then that once the serology test templates go out, then we should start going ahead and preparing that EUA through the serology template?
So you can email still to the template's email address and ask what is required for serology tests today, now. However, those templates will be made public in short order and will provide much more clarification on our recommendations for validation of those tests.
Wonderful. Thank you so much. I really appreciate it.
Uh-huh.
Next question is from Tom Hayhurst. Your line is now open.
Yes, thank you very much for your transparency in these weekly meetings. I appreciate it, as everybody else on the line I'm sure do also. I'm specifically addressing the serology testing, and even more specific, the lateral flow test. You've opened up the call with a very nice pathway forward for validation for the EUA and the submission guidelines for the lateral flow test as a device. But has there been any consideration of looking at digitizing those results through a device that may be agnostic to the vendor to pick up those lateral flow tests and enable epidemiological review of the data and other sorting of that data? Has there been any pathway forward for a device validation instrument device?
So, yeah. So we're totally open to such devices. There are already such devices for other respiratory diseases on the market that allow the recording of non-identifiable information and uploading for public health surveillance type activities. So we're totally open to that, and as long as the technology works, is accurate, we'll authorize that. There's a number of different ways you can go about that. I would just state, though, that there could be considerations or concerns about things that can otherwise be visually read rather than, say, read by some other means like fluorescence, that are where the detectors are measuring something other than a visual read. And there's things like lighting and how dark the bands are and things like that, and whether or not the device you're using to record the result is uniform, harmonized in the marketplace. So an app-like application might be very valuable, but if it's able to utilize a number of different devices, different smartphones. If the camera performance, for example, doesn't have the same resolution, there's differential ways of handling lighting, things like that. So our expert team would evaluate the technology that you're interested in developing and using in this way to make sure that an accurate result is recorded by such a device.
Very good. Thank you very much.
Next question is from Brad Fox. Your line is now open.
Hi there. Some of your information on the swabs have been answered, but specifically I'm referring to the CDC recommendation for which type of swab should be used, the NP swab or an NS swab. And does the FDA have any thoughts or recommendations, especially as it might apply down the road for asymptomatic people? For instance, large organizations which will want to test people that are asymptomatic before they return to work. So differences between the two swabs. Is there an FDA preference? The CDC recommendations is that they would prefer the NP over the NS. Any thoughts on that?
So first of all, I would defer to those kind of recommendations as far as what the CDC perceives as being important to the CDC. I think at the same time, we all realize that performing, say, depending on the interval, a weekly NP swab on people who might be considering go back to work, as some options are being considered out there, I've heard about them, could be challenging from a number of standpoints. One is that NP swabs may not be so readily available because of supply issues. The second is anybody who's had an NP swab will know, and I have as a volunteer, they are very uncomfortable, and doing that on a weekly basis could be challenging to people. So, this is an extremely challenging question. What is the about testing asymptomatic people, and in certain situations like return to work, which obviously is a very important question. Exactly how this should be done is still an unknown scientifically. Is a nasal swab as effective as an NP swab? I think more data could be collected on that, and we can see. I guess from our standpoint, if a claim is going to be made, we'd want that claim to be supported by data for one type of swab or another. Exactly how labs utilized their tests for this situation is perhaps something that the FDA may not specify unless they're making a claim. But we are working on a communication. It may just be an update to our FAQ page about this specific topic. So we'll be as transparent and forthcoming as we can, but we realize, and I hope you realize, that this is a very challenging topic.
Yes, I really appreciate that. Some of the new tests that have come out are really based on NS swabs, and I was just curious if the FDA has been doing effectiveness testing between the two types of swabs, or if that is something that would be done in the future.
So there are some studies that have been done out there showing the differences between nasal swab, mid-turbinate, and NP swabs. And then there are some developers who have done some of that work as well. Going forward, as we know that there are now patients available to do these studies, comparisons between the different swab types, sensitivities for detection would be important. I think it's generally wise, if you're going to do anterior nares, that you swab thoroughly both sides of the nose. That if you're going to do studies, be sure that you properly randomize how you swab for, say, comparison testing between different devices, different tests, different molecular tests, or perhaps to go forward, potentially direct antigen tests. And that if you also obtain another sample type like mid-turbinate or nasopharyngeal, you also take that into account as you randomize the sample collection. So you don't always take the nasopharyngeal swab first. You randomize whether you do that first versus another site like the anterior nares. So I don't think we have definitive evidence yet about how much, say, performance you might lose going from a nasopharyngeal to a nares, but even bilateral nares. The United Healthcare Group study was pretty well done, and but the numbers maybe, could be added to with other studies. But it did show that there is a potential small drop-off in sensitivity when you go from nasopharyngeal to mid-turbinate to nasal swab.
Okay, thank you so much for, it is a challenging question, so thank you for your comments.
You're welcome.
The last question that we have time for today is from Kevin Boykin. Your line is now open.
Hey, Tim. Thanks for holding these calls. They've been very informative. I work for a specimen collection kitting company, really focusing on getting supplies out to clinicians for the collection of these samples. And we're trying to address the shortage in media. And I know you guys have had some conversations around how you can address some of this other than what's been on the FAQ page. But has there been any talks around issuing an EUA or fast-tracking 510Ks for additional media that could be used for the collection?
I think we would be open to an EUA. We have been working with certain entities who have come forward to us on plans that they have to manufacture VTM and distribute it. So, some of them are, say, copying the CDC formula, and they've come forward to us, and we've worked with them on a plan to do that, and have allowed them to do that. One of the important features of VTM is sterility, whether it be VTM or whether it be UTM or, say, phosphate buffered saline. Sterility is an important factor. We realize this is a great unmet need, and we're thinking about what we might do to make greater clarification to the community on this. But for now, you can simply send us an email to the template's email address, and we will work with you one-on-one to address any concerns that we have about what you might want to do.
All right. Thank you, Tim.
And now I'd like to turn the call back over to Ms. Irene Ahir.
Thank you. This is Irene Ahir, and we appreciate your participation and thoughtful questions. Today's presentation and transcript will be made available on the CDRH Learn webpage at www.fda.gov/training/CDRH Learn by Monday, May 4th. If you have additional questions about today's presentation, please email cdrh-EUA-templates@fda.hhs.gov. As always, we appreciate your feedback. Following the conclusion of today's presentation, please complete a short 13-question survey about your FDA CDRH virtual town hall experience. The survey can be found at www.fda.gov/CDRH webinar immediately following the conclusion of today's live discussion. Again, thank you for participating. This concludes today's discussion.
This concludes today's call. Thank you for your participation. You may disconnect at this time. Speakers, please stand by.